Science, Universitas Islam Indonesia. We cordially invite you to open the camera and use the background image that has been given by the committee, and also rename the participant's name with name_agency. Once again, we cordially invite you to open the camera and use the background image that has been given by the committee, and also rename the participant's name with name_agency. Because in a few minutes, the opening ceremony will proceed in no time. Thank you. Assalamualaikum warahmatullahi wabarakatuh. To the Honorable Dr. Levi Weldron, from the Associate Professor of Epidemiology and Biostatistics at the City University of New York, the respectable the Head of Statistics Department, Faculty of Mathematics and Natural Science, Universitas Islam Indonesia, Dr. Edi Widodo SSI MSI, or the representative, Honorable Dr. Rahmatul Fajriyah MSI, as the coordinator of SBRC Webinar Series 5, and all the lecturers and staffs of Statistics Department, Faculty of Mathematics and Natural Science, Universitas Islam Indonesia, SBRC team, and also all the participants and invited guests that we respect. The honorable ladies and gentlemen, good evening and welcome to our big event, Statistics and Bioinformatics Research Center Webinar Series 5 with the topic Towards Microbe Set Enrichment Analysis for Human Microbiome Studies. This event is held by Department of Statistics, Faculty of Mathematics and Natural Science, Universitas Islam Indonesia. I am Zahra Maharani, and it is an awesome and precious change for me to be your master of ceremony in this evening. So before starting our agenda, please let me explain the schedule of our event today. The first one is opening, continued by welcoming speech, prayer, webinar from Dr. Levi Weldron with the topic Towards Microbe Set Enrichment Analysis for Human Microbiome Studies, question and answer, and last is closing. So without any further ado, let us open this event by saying Basmalah together. Bismillahirrahmanirrahim. Thank you. We hope that God will give us his blessings into our event today. The honorable ladies and gentlemen, step into the following agenda, which is the welcoming speech that will be delivered by the head of Statistics Department of Faculty of Mathematics and Natural Science, Universitas Islam Indonesia, that will be presented by the Secretary of Statistics Department, to the respectable Mr. Muhammad Hasan Sidik Kurniawan, MSc. The screen is yours.
Okay, thank you very much, Ms. Zahra. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay, good afternoon to our honorable speaker, Professor Levi Weldron from the City University of New York, and all of the committee of this webinar, and also all participants wherever you are. Okay, thank you very much to every one of you for being here today at this good moment. Although it is still in pandemic period, I hope all of you are in good health. My name is Muhammad Hasan Sidik Kurniawan, and it is such an honor for me to speak on behalf of the Department of Statistics, Universitas Islam Indonesia. Let me begin by giving you a warm welcome to this SBRC webinar series number 5. Before we get started, let us say our gratitude to Allah Subhanahu wa Ta'ala. Because of His blessing, we can meet in this event, although it is still in pandemic situation. Also, I would like to express my appreciation to the Statistics and Bioinformatics Research Center, SBRC, who generously make this webinar come true, especially to Ms. Rahmatul Fajriyah, Ms. Dinatri Utari, and also Mr. Muhammad Muwajir, and for all of our students who help us in this very event. We could not have done it without all of you. Okay. In today's webinar, we would learn about analysis for human microbiome studies. From the references I have read, microbiome is the full array of microorganisms that lives on and in human body. Specifically, we can say that microbiome is the collection of microbial genomes that contribute to the genetic portrait of human. And also I read that the microbiome has so much advantage to our body. One of them is controlling our immune system. In this pandemic situation, immune system on human body being told everywhere and every time. And I think it is necessary for us to know more about our body, especially about the microbiome's role concerning with our immune system. Hopefully, with so much research, studies, and webinar like this about this topic, all of us can give a contribution to end this pandemic situation so that our life can get better. Just for your information, SBRC is actively held this webinar series since the pandemic period began with variety of topics on flora, fauna, and also in our human body. So I think it is important that this webinar also similar research and studies like this can go on continuously in the future. So we will know more about our body, also about the microorganism living in our body and its advantage. So I appreciate to all of my friends in the SBRC team for holding this event. Okay, talking about studying our bodies and what's on it. In the Holy Quran, chapter 51, verse 20 to 21, Allah Subhanahu wa Ta'ala said, A'udzu billahi minasyaitonir rajim, wa fil ardi ayatul lil muqinin wa fi anfusikum afala tubsirun, which is there are countless signs from our Creator on earth for those who with sure faith, as they are within ourselves. So can you not see? That first ensures that in our earth and our very body, there are much magical things that happen as the signs from our Creator. And we are allowed to study it so that our life can get better. So I think it should be our motivation to know more about our body and what's on it. Well, once more on behalf of the Department of the Statistics Universitas Islam Indonesia, I would like to say welcome and enjoy your time studying to this material. It is great to see so many of you here, although it is only via Zoom. Thank you very much. Hope all of you have a good health. Assalamualaikum warahmatullahi wabarakatuh.
Thank you for the introduction. And
Okay.
it really is miraculous. I am actually in Italy right now, even though I work in New York because I was here on sabbatical when the pandemic started, and I just stayed. So I've been in Italy for almost 2 years now, in spite of my work being in New York. I'll be returning in August, but so here I am in Italy giving a presentation to a group in Indonesia, something that never could have happened a few years ago. So I'm very grateful for the things we've learned to allow this kind of meeting to happen, and to my hosts for inviting me here today.
Okay, you're welcome. Thank you very much, Miss Prof. Levi, for coming in this event. We really appreciate it. Okay.
Thank you. So let me see if I can get the screen share working properly. Are you seeing my title slide?
Yes.
Okay. So
would you like to start now?
Oh, is now a good time to start?
I think the Master of Ceremony would like to have like praying first.
Maybe,
maybe,
sorry.
After that, maybe we can start.
Okay.
Okay, yeah. Mbak Zahra, monggo.
Thank you to Dr. Levi and also to Mr. Muhammad Hassan Sidi Kurniawan that already delivered the speech. So, ladies and gentlemen, before we go to our main session, which is the webinar, I think we can first deliver our prayer, and I will invite Mr. Muhammad Robi Hada Akbar to lead our prayer. To Muhammad Robi Hada Akbar, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, let us bow our head for a moment, pray to God Allah Subhanahu wa Ta'ala, so that our event, the 5th SBRC webinar series, Universitas Islam Indonesia 2021 goes well. Al-Fatihah. A'udzubillahi minasyaitonirrajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Ar-Rahmanirrahim. Malik yaumiddin. Iyyaka na'budu wa iyyaka nasta'in. اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين. حمد شاكرين حمد ناعمين حمدا يوفي نعمه ويكافئ مزيده. يا ربنا لك الحمد كما ينبغي لجلال وجهك الكريم وعظيم سلطانك. Allahumma sholli ala Sayyidina Muhammad wa ala ali Sayyidina Muhammad. Allahumma ghfir lana wa li walidina wa lil muslimina wal muslimat wal mu'minina wal mu'minat al ahyahi minhum wal amwat innaka ala sami'un qaribun mujibud da'wat ya qadiyal hajat innaka ala kulli syai'in qadir. Rabbi shrah sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Oh Allah, today, Saturday, July 31, 2021, in this beautiful place, we gather here to bring about the 5th SBRC webinar series, Universitas Islam Indonesia, with the topic is Toward Microbe Set Enrichment, Analysis for Human Microbiome Studies. We are begging you, Allah, to make this webinar as a useful science assembly, as a medium of sharing useful ideas, knowledge, and experiences of scholars, researchers, and students of various disciplines. May the webinar we organize today benefit our life, broaden our knowledge, shine our ideas, and lead us to be successful, productive person, which in turn will boost the dignity of our nation. Oh Allah, guide and bless our heart and our minds with the light of their guidance. Impart your supreme wisdom upon our activities. Help us to speak our minds clearly. Help us to listen to each other, respect each other, love each other, so that we are included to the blessed person. Oh Allah, protect us from unintended temptation. Show us the right path and give us knowledge and strength to perform good things. Equally, show us and make it clear the bad things and give us knowledge and strength to avoid them. Oh Allah, you are the one who can fulfill our dua. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Subhanaka rabbil izzati amma yasifun wa salamun ala mursalin walhamdulillahi rabbil alamin. Amin, 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 ya rabbal alamin. Thank you for your attention. Now back to the Master of Ceremony, please. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for Muhammad Robi Hada Akbar for leading our prayer. So the respectable ladies and gentlemen, before we start our main session, let us have the photo session first for the documentation. So we cordially invite all the participants to turn the camera on and use the background image that are already given by the committee in the chat column. Once again, we cordially invite all the participants to turn the camera on and use the background image that are already given by the committee in the chat column, because we will have a photo session. Once again, to all participants, please to turn the camera on and use the background image that are already given by the committee in the chat column. Thank you. Okay, I think we can start right now. I will count while we are preparing our best smile, okay? One, two, three. Yes, once again. One, two, three. Okay, thank you. And the excellencies, all of the participants. Now, finally, we come to our main session, the agenda that we already waited for, which is the SBRC webinar series 5 with the topic Towards Microbe-Set Enrichment Analysis for Human Microbiome Studies. So without any further ado, we would like to welcome Dr. Levi Waldron from Associate Professor of Epidemiology and Biostatistics at the City University of New York that will be our speaker in this evening. And he will be assisted by Dr. Rahmatul Fajriyah MSE, who will be the moderator. So to the respectable Dr. Rahmatul Fajriyah MSE, the time is yours.
Okay, thank you very much, Mbak Zahra. Good afternoon and also good morning for Professor Levi Waldron. Today, I'm being your moderator for his talk. His talk is about, wait a minute, this one. So the title of his talk is about Toward Microbe-Set Enrichment Analysis for Human Microbiome Studies. And then before we start, maybe I would like also to, how to say that, to introduce about him. Dr. Levi Waldron is an associate professor of epidemiology and biostatistics at SUNY Graduate School of Public Health and Health Policy in New York, with research interest in cancer genomics and also metagenomics. He is a long contributor to an organizer for the bioconductor projects for open source bioinformatics, with an interest in creating well-documented and user-friendly methods, data structures, and databases that help find new value in published data. You can see as well about the abstract of his talk in here. I think it is time for him to start his talk. Thank you very much. You can start your talk now. I will stop my sharing. Professor Waldron, the time is yours.
Okay, thank you very much for the kind introduction. We'll try. Okay, can you see my presentation now? Okay, great. Thank you everyone for coming. I was told that this is primarily a statistics audience, but also a fairly diverse audience with people in different areas of study. So I've tried to make this a fairly generally accessible presentation, but one with that should have some points of interest for a statistics audience. When we look at a person, we tend to see a person like on the right. This is a famous picture from the cover of Nature about 10 years ago. But the point that this picture is trying to make is that as we look closer, we may begin to see something different, which is a large collection of diverse microorganisms that inhabit every part of our body. And these organisms vary greatly from different parts of the body. So the types of organisms and the physiologies that are required to inhabit different parts are different. But as you can see from some of these pictures, they look completely different. It's really an incredible diversity of microorganisms that inhabit us. And these are only bacteria. And in fact, we're inhabited by bacteria, archaea, eukaryotes or fungi, and viruses. And it was really only in fairly recent times, in about the past 10 years or so, that we started to understand just how diverse and extensive this community was. The microbiome has been studied for a long time now, for 100 years or something, but only since the advent of sequencing did we realize just how many cells, how many microbes, and how much microbial diversity exists in each one of us. And we're now able to start asking questions like, who are they and what are they doing? And our main tool for doing this is metagenomics, which is the study of uncultured microorganisms from the environment. That is, we take samples directly from the spot. We do library preparation to extract DNA from this entire collection and just sequence this DNA from whichever organism it came from. So unlike sequencing of the human genome, where we're focusing only on one genome, we can be sequencing genomes from dozens or hundreds or even thousands of different organisms. And in doing that, we focus both on taxonomic, that is, who they are, and functional characteristics of this total collection of microorganisms. Our knowledge of the importance of host-associated microbiota is not new. I put this slide here mostly because it was interesting to see that as far back as 1975, it was known from studies on lab animals. No biotic are animals that are raised without being allowed to be colonized by any microorganisms at all, so that they could be studied experimentally. And all of these were things that were known to be roles of the host-associated microbiome, from synthesis of vitamins and other digestive and metabolic processes to competition with potential pathogens. So simply having the presence of a healthy microbiota can prevent pathogens from being able to establish themselves. And as one of our hosts mentioned, they facilitate a normal immune system function. And more recently, these are just in the past 10 years or so, there has been an increasing evidence of their importance in human health. Although this has been assumed and understood to be true, there has been limited ability to actually make, I wonder why I'm getting some drawing on the screen. Maybe that's not me. I thought it was something I'd accidentally put on my slide, but I don't think it's me. Anyways, there have been some success stories of actually modifying the microbiota of individuals to treat various illnesses. So the most well-known example is the treatment of antibiotic-resistant C. difficile infection. So this is a bacteria which certain strains can cause ulcers and ongoing gastrointestinal distress. And these certain infections have been treated repeatedly with antibiotics, and the same C. difficile keeps on returning and causing problems. But there have been some great successes in these cases of combining antibiotic treatment with a transplant of fecal microbiota from another host with a healthy gut. And this has been some 90% effective in treating cases that were previously untreatable. There's also some preliminary evidence that fecal transplantation can cause mice to be lean or obese, depending on whether their microbiota are coming from a lean or an obese person, and that it may also modulate resistance to immunotherapy in cancer. So all of these are based on fecal microbiota transplants. And so this is these provide evidence that the microbiota can affect these outcomes, even if we don't understand why or what complex changes are occurring during these transplantations. So I'll talk a bit more now about the methods and how we can study the human microbiome now. And as I mentioned, our main tool is metagenomic sequencing, where we have this mixed microbial community with many different microbes present. We undergo some lab procedures to extract DNA from all of them. And this is just an example of 4 different genomes. And then there are 2 main classes of methods that are used. One is called amplicon sequencing, where we just select a single gene that is present across all bacteria or across all eukaryotes that is fungi, and sequence that one single gene in order to infer which species or which genera are present. And this has been the major method used. It was the first one to gain widespread adoption because it's relatively inexpensive. You don't have to do very much sequencing to gain data like this. So if you were to look in the literature right now at how people have been doing metagenomic sequencing, you would see a majority that would fall under amplicon sequencing. And if you're looking at the paper, you'll know this from keywords like 16S, 18S, or ITS. And this refers to a single gene that is present across all species being studied, and that is the gene that is being sequenced. The other major category is called whole metagenome shotgun sequencing. And in this one, no single gene is isolated. The entire genome is broken into fragments and sequenced. So this requires much more sequencing. And in the earlier days of microbiome sequencing, this was very expensive. It was understood to be superior because you get more data. You can do more different things with it. You can identify individual genes that are present. You can identify with greater resolution which species or even strains are present, but it was expensive. This difference in price now between amplicon sequencing and metagenomics sequencing is a lot less now than it used to be. And in fact, this DNA extraction methods is now more expensive than either kind of sequencing. So metagenomic shotgun sequencing is much more widely used now than it was previously. And it may, in fact, be taking over amplicon sequencing in popularity because it gives so much more and high resolution data at a comparable cost. So just for people who have never analyzed microbiome data, to give you some idea of the general strategies that most analysts approach it with, one and probably the most important and widely used method is actually the simplest, which is just to treat each measurement separately. So if we find that we have 100 species present in a pool of samples, we just look at each species as a single variable, and we use in fact common tests from classical statistics. The non-parametric tests such as a Wilcoxon test are popular, or sometimes parametric tests, but there is nothing special about this. It's really just analyzing one at a time, followed by a correction for multiple hypothesis testing if you are doing hypothesis testing. There are, of course, a lot of refinements of this for so-called high-dimensional data where we have many variables. There are Bayesian methods that borrow information on variance to help stabilize these tests. But they're fundamentally no different than the univariate tests. A second major method is dimension reduction through principal components analysis and related methods, some of which use distance metrics that are more specific to ecological data. But this is also a popular and I would say a good exploratory data analysis approach. The third is metabolic pathway inference, and I'm not going to talk a lot about this, but I wanted to make you aware that that it exists. And the idea behind this, unless we're actually directly measuring the compounds produced in metabolic pathways, which usually is not done, we don't actually have this data. All we have is DNA sequences, and so in general, the approaches are to take those DNA sequences and relate them to known genes and proteins and try to infer what metabolic pathways are present in the DNA and have the potential to be active, even though it says nothing about whether they are active or not. But there are differences in the metabolic potentials of different microbes, and so it is possible to do some inference on the potential. And then the final category of common analysis is ecological diversity. And this is a great simplification of the data because we take all of these data and just calculate one single measure about how diverse the sample is. So the simplest ecological diversity measure would just be a count of the number of species that are present. So a sample with 2 species present is less diverse than a sample with 100 species present. Some alternatives to this also account for the evenness of diversity, so that 2 samples that both have 100 species present, the one where they're all present in equal abundance is more diverse than the other sample where 1 species is dominant and all the others are present in small amounts. This metabolic pathway approach has had some promise because there is the assumption that so on the top here, I'm looking at taxonomic inference. These are phyla, which is the highest level of taxonomy. And so if 2 species are members of different phyla, then they're very, very different from each other. And this is a plot of the different phyla that are present in the nasal, skin, oral, stool, and urogenital tracts of humans from the Human Microbiome Project study. And these are 1, 2, 3, about 10 different phyla. And I just want to point out that this is pretty amazing. So looking at the stool microbiota here, this green spot, these are firmicutes, one common phyla, and the blue here are Bacteroidetes. And so these are healthy young volunteers. They're all in their 20s. They're all healthy. They all live within a couple regions in the United States. So this is not a very diverse sample, and yet this is an incredible diversity of microbiota just within a single body site. We vary between having about 95% Bacteroidetes to maybe about 80% firmicutes. And just to give you an idea of what it means that these are different phyla, if we consider the phyla that humans are in, if we go to another phylum of animals, we would be looking at something like mollusks or insects, invertebrates, things that are very, very different. So these are extremely different bacteria, and yet they can be found in very much different abundance, even within healthy samples. We do see, however, that when we infer the presence of metabolic pathways, there is more consistency between individuals. And a lot of that is because regardless of what species or even phyla are present, they all have to perform certain basic functions in order to survive in this environment. And so that raises the question of whether the analysis of metabolic pathways is useful, because it's interesting to see that it's conserved between people, and looking at healthy people, the hope here was that this would provide a baseline for what is required to be healthy, and that we would see changes in this in individuals with disease. But I think that in general, that has not been the case, that we continue to see consistency in the metabolic pathways also in people with disease, and that it may in fact be the individuals who is there that is more relevant. That's a very broad statement to make, but analysis of taxonomy remains far, far more common than of metabolic pathways. Looking at that within sample diversity that I mentioned, the simplest example being species richness, we see that also varies greatly even between healthy individuals. And this is another example from the Human Microbiome Project, this group of very healthy people from only a couple parts of the United States. And we can see that between body sites, it varies a lot. So there's much less diversity in the urogenital tract than there is in stool or in oral sites or nasal sites. But there's also a lot of variability within individuals. So this is variability of let's see, log 2. So this is a factor of 2 to the 4 difference in species diversity just between individuals in the same site. So that's some 16-fold variation in diversity. So all of this makes for a great challenge for understanding these systems and for the statistician. And then to complicate things further on top of the underlying complex biology, study design is a challenge. And as someone who has spent a lot of time studying the human microbiome, what you will find is a lot of samples of convenience and almost entirely observational studies. So in these kinds of studies, confounding and reverse causation are always issues to be aware of and to try to identify threats to the validity of the study. And one instance where confounding or reverse causation can be important is antibiotics usage, because antibiotics usage can have a profound effect on an individual's microbiome, decreasing the diversity and changing the composition. And it's also related to overall health, in that generally unhealthy people are more likely to be administered antibiotics. And it may in fact be a treatment for the disease under study. So we can have instances where we are studying a particular disease in a case control type design, but the people in the case group have been repeatedly administered antibiotics over years, and their microbiota have been affected by the treatment for the disease that we are studying. Additionally, power and sample size are a big issue. These are high dimensional data sets, so we have probably hundreds of variables, and yet we often have only dozens of samples in the study. Furthermore, batch effects are important not only to human microbiome studies, but to all kinds of measurements. And in fact, I have made the point that what we call batch effects in bioinformatics and the study of genomics has been known for long before these, under other names such as interviewer effect or measurement bias, in the same way that if we have two different researchers administer a verbal survey, we may get different results from participants depending on which reviewer asked the questions and on the way that they ask the questions. And similarly, if we have two different laboratory technicians performing an assay, they may perform it slightly differently and get slightly different results consistently, or even the same laboratory technician performing the assay, but on different days or different months using different batches of reagents. All of these can cause a consistent bias in results. This was a landmark paper that really brought the issue of batch effects in all kinds of high throughput data to the forefront of people's minds in thinking about study design and analysis. And I think the main thinking about batch effects now, certainly the way I think, is that they're impossible to avoid, but what you can avoid is a situation where your batch effects are confounded with your treatment. So here's an example of where we have two different batches of reagents, and one was used a little bit beforehand. We're looking at a time period over time, but after a certain time, as the first batch of reagents ran out, the second batch started being used, and the abundance of Microbiota, a genus, increased using this second batch of reagents. So this is okay as long as these batches are not confounded with the outcome that we're interested in. So if we studied all of our cases in batch 2 and measured all of our controls in batch 1, then there's a serious problem and there's really no way to recover from that in the analysis. But as long as we planned for this in advance and used locking and randomization to spread our samples randomly and in a balanced way across the batches, then we can actually correct for batch as a nuisance variable, as a covariate, and there are plenty of tools for doing that. And even if you didn't correct for batch, you would just have an unwanted source of variation in your data that could be corrected for. But the result of all of these is that it can be difficult to interpret results of studies, both because of their high dimensional nature and because of the potential for threats to their validity. And it can be difficult to compare between published studies, which use different methods and different ways of reporting their results. So with that rather long introduction, just to orient you to the field, here's an outline for the rest of the talk. I'm going to start with two examples, and I'm going to make the case for the work that I am doing through these examples, and they will show you how there's a need for microbe set enrichment analysis in these situations, and I believe many more. And then I'm going to show 3 different projects of ours relating to this work. So my first example comes from an analysis of the New York City Health and Nutrition Examination Study. And this is a study that I was involved in the analysis of. And this was a sub-study of the tobacco-associated saliva microbiome. This was published a couple years ago in the Annals of Epidemiology, along with a second paper on sociodemographic variation in the oral microbiome with collaborators from the University of Trento here in Italy, where I have been. So this was a population representative study of adults in New York City conducted by my school and the Department of Health, and a sub-study that was initiated by my colleague, Professor Heidi Jones. And in this sub-study, we we took a subsample to create a case control study where we contrasted current cigarette smokers And the current cigarette smokers were confirmed in 2 ways. The first was through a questionnaire where people responded to a question about their smoking habits. But the second was that we actually had blood samples from all of these participants. So we were able to observe the presence of a metabolite called cotinine, which is a byproduct of the metabolism of nicotine. So for someone who has no exposure to nicotine, they will have no cotinine in their blood. So we were able to confirm both through questionnaires and through the presence of cotinine in the blood that these were current cigarette smokers or people who reported having never smoked and also had no cotinine, no byproduct of nicotine in their blood. And we selected, this N equals 86 was out of about 1,500 individuals in the study, and these were the ones with the highest levels of serum cotinine in their blood. So these are individuals who are very heavy smokers. This plot is a differential abundance analysis. So this is following that first method that I showed you of just univariate analysis. This DE-seq2 is one of those Bayesian adaptations that I mentioned that borrows estimation of variance across all the variables in order to provide some smoothing and reduce false positives. But it's essentially, this is a negative binomial log linear regression for count data. And doing this, we identified these are genera, so that's the taxonomy level 1 above species, and they're categorized into phylum here. And we identified several genera from the Firmicutes phylum that are higher in smokers, and then several mostly from the Proteobacterium or Proteobacteria phylum that are lower in smokers. So that is fine. We have some results, but these are difficult to interpret. Probably none of these mean anything in particular to any of you. So if this were the end of the study, you would say, okay, well, maybe smoking alters the oral microbiota. I'm not sure if I'm convinced. Maybe they're false positives, and I don't know if it is biologically significant or plausible. Oops. My slide changes are very slow for some reason. Okay, so we wanted to say not only that there were certain bacteria that were differentially abundant between the saliva of smokers and non-smokers, but we actually wanted to try to infer whether these differences were in fact caused by smoking and not just some incidental differences. And we did this in several different ways that I'm going to show you just because I think they're a very interesting application of causal inference that would be of interest both to a statistics and a medical audience. But the first of these is a very standard approach to causal inference, which is simply to fit a multivariate model where you adjust for hypothesized confounders. So we've added age, sex, race, ethnicity, self-reported physical activity, education, diabetes status, and self-reported gum disease, all as potential confounders. And what I'm plotting here is for all of our variables. These are 212 different variables here. These are the regression coefficients in the crude analysis with only tobacco exposure as the predictor, and in the adjusted multivariate analysis. And what we see, and these in gray are the ones that were not significant in the crude analysis, and the ones in black were statistically significant in the crude analysis. And what we see is generally a relationship along the identity line, where the adjusted coefficients are approximately equal to the crude coefficients. You see at the very high end here some movement towards the null. These are some coefficients that are less significant or have a lower adjusted fold change than crude fold change. That said, some of these are probably not confounders in the sense that we mean them as biostatisticians and epidemiologists, in that I think self-reported gum disease likely does not cause smoking. That relationship is probably the other way around. So in some of these cases, I might be more inclined to believe the crude analysis than the adjusted analysis if smoking is the main change. But this is only one line of evidence. To make causal inference from an observational study, you really need multiple lines of evidence to begin to believe the causal effect. So the second approach to causal inference was to compare the effects of smoking to the effects of secondhand smoke. And we were able to do this because we had serum cotinine levels. And so we were able to identify people who reported on their questionnaire that they were non-smokers, but nonetheless, they had serum cotinine present in their blood. And so we performed a crude regression with these individuals with the quantitative level of cotinine as the predictor, and then performed another regression on the coefficient, so a regression of regressions, to identify a reasonably strong correlation. While 0.4, that's pretty high in the world of high dimensional data analysis, and one that was highly significant between the coefficients of these two analyses. So that means that the bacterial genera that were increased in smokers tended also to be increased in the individuals exposed to secondhand smoke, and the ones decreased in smokers were also decreased in individuals exposed to secondhand smoke. So this is a fairly strong piece of evidence for a causal effect of tobacco exposure, because these are entirely separate groups of individuals and entirely separate measurements and scale, and yet we see this consistency in the effect of tobacco. And then the third part, which is the one that gets to the title of my talk, towards microbe set enrichment analysis, was an attempt to understand biologically what is happening to the people with exposure to secondhand smoke or to smoke. And this is an analysis of what I call a microbe set. That is a group of microbes that in this case are anaerobic. And this is a bit of an unfamiliar plot if you have not been in genomics and studied gene set enrichment analysis. But what this is these lines along here represent all of the anaerobic bacteria out of about 400 genera that were present. And this shows the log fold change. So these are the ones that are decreased, and these are the ones that are increased. And what we're looking for in this analysis is whether this group of anaerobic bacteria tends to be clumped towards one side or the other here. And there are statistical tests that can be performed to assess whether this distribution is random or focused towards one side. And in this case, it is strongly focused towards one side. And what that suggests is that tobacco smoke affects aerobic microbes and the balance of aerobic and anaerobic microbes. And this decrease of aerobic microbes means that in the saliva of smokers, there are fewer microbes that need oxygen and more microbes that do not need oxygen. So this helps provide some biological interpretation for those lists of genera that we saw, and helps to make some sense of it. And it also helped us to have some understanding of where to look for lab-based evidence. And we were able to find that there have been experiments done just in cultures showing that exposure of microbiota in a petri dish in the lab to tobacco smoke will tend to have more of an effect on bacteria that need oxygen than on those that do not need oxygen. And that brings us to my second example, which is more of a statistical analysis and a collaboration with another group in Italy that I did while I was here. And the objectives of this study were this came also in time after the first study that I showed you. And I was really impressed with the ability of microbe set enrichment analysis to help us understand differential abundance in the smoking study. And I thought that this was something that we should also be using to benchmark differential abundance methods to tell us not only can they control type 1 error rate, et cetera, but do they provide us with biologically meaningful results? And we addressed some key questions in this paper. One of them, which is argued about a lot, especially among statisticians who study the microbiome, is that microbiome data are probably compositional in that all we can ever measure is the composition of species or genera. That is, what proportion of the total that they compose. We can't make absolute measurements, and so there are specific methods for compositional data analysis that account for this relationship between all species present. But there is a question, there are always trade-offs in any analysis method that we choose. This means that we cannot treat them as independent variables like in that analysis that I already showed you. It limits you to certain other subsets of methods. So the question is how important that is and whether it outweighs some of the gains that might be made through making assumptions of independence. And then there are a bunch of other questions about differential abundance. Do they fit the data well? Do they control type 1 error? Do they have good power? Do they produce concordant results in independent samples? But especially this final one, do they produce biologically meaningful results? And I argued that this is a question that, while it is the most important to medical and biological scientists studying the microbiome, it has been completely missing from benchmarking studies of the human microbiome. And so I took a data set from the human microbiome study where I thought that this difference between aerobic and anaerobic bacteria should be very strong. And we have in there observations from the same individuals in the supragingival plaque just above the gums, and from the subgingival plaque taken from underneath the gums. So in the supragingival area, there is oxygen available. They're directly exposed to the air in your mouth. And in the subgingival plaque, this is a hypoxic environment. There's very little oxygen there. So we really would expect a difference in the oxygen utilization. And some of this is already known from laboratory experiments. There's a difference in the, has to do with the cell membrane, with the shape, and in fact also with oxygen utilization. So in addition to assessing those other properties, statistical properties that I mentioned, we also assessed the biological meaningfulness of the results. And this is this bar plot. I'm showing here the salmon colored bars. This is a good result. This is a this is an obligate aerobe. So this is a bacteria that needs oxygen, and it is overabundant in the supragingival plaque. Then the blue bars that go down, this is also good because this is an obligate anaerobe. It can only survive in an anaerobic environment, and it is down in the supragingival plaque. So these are what we would expect based on what we know about the oxygen in these environments. And then the greens, these are bacteria that can be either. They can metabolize with or without oxygen. And we see a very big difference between these results. So some of the best methods, let's say this right here, edge R with trimmed means of M values, this stands for. This is a method just borrowed directly from RNA sequencing, and it performs pretty well. It identifies a lot of anaerobes that are down in the supragingival plaque, and it also identifies quite a few aerobic bacteria that are up in the supragingival plaque. But we see this MSIG, MGS ZIG, this is a zero inflated Gaussian method that was proposed specifically for microbiome data analysis. This has some problems. It did well for the aerobic bacteria, but it actually did terribly for the anaerobic bacteria. It identified equal numbers as being up and down in both environments, which is not plausible. And then over at the other extreme, these are also some microbiome specific methods that seem to just have very, very poor power to detect anything, even though we know that there should be a difference in these groups in this comparison. So I thought this is a very interesting comparison of not only the statistical properties, but how it performs in a situation where we know we should be able to find some results based on what we know about these environments. And this analysis is based on a particular cutoff, but we also looked at a cutoff-free method. So that last one was based on a false discovery rate of, I think, less than 0.05. This is looking across all different thresholds. So the methods that had poor power don't necessarily suffer here because we just consider a threshold based on the percentage of the taxa that we consider to be predicted as differentially abundant. So there's no power consideration here. But across this entire range, we see some very superior methods. These are ones, this is the true positives minus the false positives based on what we expect from the biology of the environment. And again, these ones at the top, this blue and the violet, these are again just methods that have been very popular in RNA sequencing. They were not developed for microbiome data. And these ones down at the bottom that have a high number of false positives relative to the number of true positives, these are again microbiome data specific methods. So there's something, there really is something wrong here, that these methods that were designed specifically for compositional microbiome data are performing poorly in a situation where we know biologically what some of the results should be. We did all of those analyses with just 3 microbe sets: aerobic, anaerobic, and facultative anaerobic. But it was a proof of concept that showed the need to collect more of these microbe sets and to start moving some of the field towards microbe set analysis as part of interpretation of these results. And anyone like me who first was studying transcriptome analysis, I started out in bioinformatics studying microarrays and then moved to RNA sequencing and then microbiome analysis, will be familiar with gene set enrichment analysis. It is a fundamental method in transcriptome analysis that that is really used in most studies that are performing whole transcriptome analysis. But these rely on databases of signatures, such as the gene ontology, MSigDB. These are large lists of genes that share some property in common so that we can study them together and see whether that group of genes is coherently changed in a condition of interest. Because otherwise it's impossible to systematically compare your results either to the published literature or to biological, physiological properties of the microbes. So this brings us to the first project that I want to tell you about. BugSigDB.org is a comprehensive database of published microbial signatures. And this has been in progress in my lab for a couple of years now and is just now approaching public release, which I'm expecting hopefully in the coming month or in the coming couple of months. I can't give a precise date yet. But the objective of this project is to standardize published microbiome signatures of different health conditions and exposures in a publicly editable semantic media wiki. MediaWiki is the same system that powers Wikipedia, so the idea is that this not only will provide microbe sets for enrichment analysis, but it will provide a platform for researchers around the world to contribute additional microbe sets in their area of interest or from their own publications and make these available to everyone and accessible for high throughput analysis. So it records not only which study a signature came from and which species or genera are in the signature, but it also records study information such as the study design, bioinformatic and statistical analysis, diversity measures, in addition to the differentially abundant taxa. And as soon as something is entered into this wiki, it becomes available for bulk data export. And we have a package that is submitted to Bioconductor. It's on GitHub right now. It will be in Bioconductor soon, called BugSigDBR, to facilitate import and analysis of these signatures in R. They're also available just as plain text files, CSV files for download from BugSigDB.org, but this makes life a little bit easier for users of R and Bioconductor. You can see on here we started in September of 2018, so it's actually almost 3 years since we started collecting data for this. We have studies from all around the world. Most of them are from the United States and from China, but we do have coverage from many countries around the world. It looks like we also have at least one from Indonesia in there. I forgot to check that beforehand, but you can go to the search box here and type in Indonesia and see what studies are represented from Indonesia in the database. We've been attempting to cover all body sites and all disease outcomes to have a very broad, diverse representation of signatures in the database. We have most human body sites represented in the database. We also have a variety of conditions: cancer, anatomical system diseases, metabolic diseases, I'll just point out from this that we use an ontology both for the body sites and for the diseases, which allow someone entering a study to put specifically what type of cancer this is, but also within an analysis in R to then easily summarize it by grouping all cancer-related signatures together or all metabolic disease conditions together. Even if you didn't know that something was a metabolic disease, this is just a function call away. We see that certain genera are very common. The Streptococcus, Prevotella, Bacteroides, these appear very frequently in signatures, but then there are a lot that only appear once. So there's a long tail of how frequently a bacterial taxon appears, and there's also a long tail in the signature size, with most of them being just 1 or 2 or 3 microbes, and a few of them being very large. This has grown a lot in the last 3 years. We actually started using a spreadsheet and now have the MediaWiki available. It now has almost 2,000 signatures from more than 500 papers, and I've had over these years more than 30 curators enter or revise data in the system already. And this is before public release, which is coming up. It's pretty interesting to look in here also at some of the differences. This is not like a random sample of the literature, but it does give you an idea that most of these studies are case control. There are a few cross-sectional studies, fewer longitudinal studies, and it also gives you an idea of when how frequently antibiotics are used as an exclusion criteria for participants. In case control studies, it's more than half. In longitudinal studies, it's less than half, because obviously you can't predict whether someone will be given antibiotics at some point during your longitudinal study, and it's less relevant to exclude them at the beginning based on it. We have various different sequencing platforms, wide variety in sample size. This is exposed or cases, and unexposed or controls, and also a wide range of multiple hypothesis testing, correction, and statistical analysis approaches. And part of the goal in collecting all of these data is to allow us to potentially identify patterns or biases in the results that are found based on the methods that are used. This is what the wiki looks like, and you can now go there. If you go to bugsigdb.org, you'll see a draft front page. There's actually right now a link that says to go to a dynamic front page, which is more interesting. That will become the front page soon in the coming weeks, and you can click on a study, either a random study at the top, or from the dynamic front page, it will also show you some random studies where you can see the experiments that were done, the signatures that were reported, and you can also look at any taxon that you're interested in. So here's an example of the Coprobacillus taxon, and if you go to its page, you immediately see all of the studies and signatures where it was reported and which other taxa it was reported along with. I added this just before the presentation here just to say that there is help wanted on this. If you're interested in this, this is a great way to learn about the field and get involved with it. I have continually curators getting involved through contributing to bugsigdb.org, and I hold weekly lab meetings among curators to discuss what they're doing. They take turns presenting a paper that they're working on, and I attend those meetings as well. We have a Slack channel to discuss issues that come up, so this is a very active area of research for me, and an interesting thing for people who would like to learn something about both about the human microbiome literature and about the methods that are used to study it, and about how taxa are named and identified as you enter these data. So, and then we have data entry. We also have review and cleanup. There's lots to be done in bugsigdb.org. And the great thing about it is that as soon as you enter something, it becomes analyzable. And there are also a lot of interesting statistical analyses to be done with this. And it's such a new resource that the data are now ahead of the methods that we have to analyze it. And we're just starting to try to develop methods for the statistical analyses of these signatures and of the methods used to develop them. So the second project I wanted to tell you about is, so that first one was focused on the literature of reported differentially abundant bacteria. This is about physiology, like the signatures of aerobic and anaerobic bacteria that I told you about. The issue here is that while many resources exist for annotating and understanding the properties of bacteria, most of them are they're both heterogeneous and they're not intended for high throughput enrichment analysis. And so our goal is to collect these physiologies in a way that allows us to understand bacterial function from microbiome experiments. And this is primarily the work of a postdoc in my lab, Kelly Eckenrode, and a high school intern who has done a lot of work helping to collect these data. Just to give you an idea of the kinds of data that we have been collecting, there are data on cell shapes, on cell wall composition, optimal pH and temperature, oxygen utilization, like I've been talking about, metabolism, what kinds of food they need, and whether or not they can move on their own. We're actually somewhat ahead of this now, but we have curated information from at least 6 databases and 15,000 different taxa on 33 different physiological features. These are not yet published, but if you are doing human microbiome analysis and think that this might be useful to you, I'm happy to share them in advance for you to try out and see if they help you to analyze your data. So in all of this process, I've discovered that curation, the standardization of these reported results, is a lot more work than it should be. And this is just an example of the many, many, many different ways that differentially abundant microbes are reported in the literature. These are all examples of where curators contributing information to BugSigDB have had to try to extract which are the overabundant and which are the underabundant bacteria. And we have everything from bar plots, box plots, tables, and heat maps and networks, everything except a simple list of taxa, which is what we need to perform enrichment analysis. And they are so complex and heterogeneous, there's no way a machine would ever be able to, I mean, knock on wood, we're certainly nowhere close to having a machine that would be able to standardize these suitably for analysis. And so my PhD student, Chloe Mirzai, has developed a checklist to try to improve how microbiome studies are reported. So it's a simple checklist that both helps authors to write a well-organized microbiome paper. It has sections corresponding directly to the sections of a paper and telling you what to put in each section, and a tick mark to say if you have done it, and comments to go along with it. And we've attempted to be agnostic of methods, databases, and analysis. So we don't say how you should do your study, we only help you to report it in a clear way. We formed a consortium that now has over 100 members from 70 institutions from all different fields of study. And this manuscript has been recently accepted in Nature Medicine and will be published in the coming months. This is just an example of what the checklist actually looks like. To conclude, I just wanted to let you know about some of the different analyst-friendly databases that I have contributed in the past to Bioconductor. Many of them start with the word curated, although not all of them. I started out in the microarray area. These are all primarily microarray data sets, curated ovarian data, colorectal cancer, and bladder cancer, which were done. These were primarily by collaborators, but that I contributed the design to. Then more recently curated TCGA data. This is the Cancer Genome Atlas and its companion package TCGA utils. These make use of a data structure developed in my lab called multi-assay experiment that integrates all of the different assay types into a single object to make it so that integration doesn't have to be done by the analyst. You can imagine that if you have 5 different assays performed on an overlapping set of specimens, this creates a very significant data management and integration challenge before you can do anything with the data. So with multi-assay experiment, we do that in advance so that you can select any subsets of the data and reshape it into the formats needed for analysis. And we've also done this for the C-Bioportal. That's the cancer bioportal that provides more than 150 multi-omic cancer data sets, also as multi-assay experiment. We have 2 packages in the area of microbiome data analysis, HMP16S data. So this is amplicon sequencing data from the Human Microbiome Project, and curated metagenomic data, which this update is actually complete now to a new version of the software that doubled the number of species in the reference databases and that are present in the data sets now, and about 20,000 samples from 80-some studies. And then our most recent paper that is not yet published, or most recent data package, is single-cell multimodal that provides selected landmark data sets from new single-cell multimodal technologies. So I'll end just with a thank you to the people in my lab who did most of this work, and to the many collaborators who have contributed to it. And I'll thank all of you for attending and for your interest. And I'm happy to stick around for a while for some questions.
Okay, thank you very much, Professor Wadron. Anyone has any questions? If you have any question, you can directly unmute yourself, or you can type it on the chat box. Hello? Okay. There is Panto. Please go ahead.
Hi, Levi.
Hello.
Thank you for very interesting. Thank you for interesting speak this afternoon in Indonesia, and I would like to know, because I'm also working with this microbiome, and in Indonesia it's just still an infant. Many people like now they know how to do it, but they don't know how to analyze it, because I think back 3 years ago, the sequencing is still very expensive in Indonesia, especially for this metagenomic. 3 years ago, one sample is around 10 million. So you can imagine what sample is 10 million. And I would like to know about the thing that you mentioned before about sample size.
Yes.
Because it's very important when we do statistically analysis, we should know the limits of the sample that we use to be really applicable statistically.
Yes.
So in your opinion, how many sample is minimal for doing this kind of microbiome research?
That's a great question. And I have to admit that in most sample size calculations that I have done, the assumptions seem completely arbitrary because they're effect sizes and variances that we do not know. So the most confident answer that I can give you is the more the better. But to give you an idea of my gut feeling, I guess, based on having analyzed a lot of data sets, is that it seems like for a lot of conditions, sample sizes in the range of even, let's say if we have balanced numbers of cases and controls, that a sample size in the range of 50 can give meaningful results.
50.
And yeah, and if there are strong signals present, that you will see it even with 25 in each group. With very strong signals, even less might be okay, but let's say maybe n equals 20. But it really does depend on how strong that signal is. If you were studying antibiotics exposure, you could certainly study that with a small sample size. If you're studying something that is much,
because it's also depend on our grant, right? Because I told you before that in Indonesia, right now, if you want to, right now it's the price is decreased rapidly. 3 years ago, still like 10 million for one sample, and now is roughly 3 million per sample. So if we really want to do microbiome analysis or microbiome study, it's really costs a lot, especially in Indonesia. Yeah. So that's why
I think, you know, with curated metagenomic data, my first approach is always to go and try to see if there's something already published that is related, where I could get an idea from something already published of what whether their sample size was adequate or not. And I think that's the best way to do it. You take their data set, you do a differential abundance analysis, see if there are some significant results, and then, you know, you can simulate a subset of their samples. That to me is the best approach to do it. When it's something that, as far as you know, has never been studied before, you just have to guess. It's an exploratory data analysis. You do.
Yeah, that's right. That's correct.
Yeah.
Because last year I did research on microbiome. I compare breast milk from pesticide contaminated area and non-pesticide contaminated area, and I used at the time 40 sample. So I use breast milk from a mother who still give breast milk fat less than 6 months, because I want to know the comparison between microbiome from pesticide contaminated area and non-pesticide contaminated area. And from that data, I know that there's a difference between non-contaminated and pesticide contaminated area, but I'm not sure about the size sample. That's why I'm still writing the manuscript about this thing.
That's a decent sample size. I think that, you know, and I mean, you will see from the statistical analysis whether you have some statistically significant results. In a question like around breast milk, to me, the main concerns that those are going to be low microbial biomass samples. So contamination is always a threat to that kind of study. So just trying to be sure that there's no, you know, that the samples are randomized across time and blinded to the person doing the study, so that if, you know, if there is a change in contamination or something over time in the lab, that doesn't differentially affect your two groups.
Mhm.
And just having controls, having like negative controls to assess what contaminants are present, and you know, efforts to minimize contamination, because it is very challenging.
Yes, exactly.
It's not like studying saliva or stool where there's lots of microbes there.
Mhm.
But yeah, your sample size doesn't sound terrible to me.
Okay. Okay. Thank you.
You're welcome.
Okay, Pak Anto. Any other participant have some question? Okay, there is one question in the chat box. Would you like to read it, Professor? Okay.
So could you please recommend the genome reference that we can use? Yes, what I recommend, what I use is a software called Metaflan, and it includes the genome references. So I don't actually download genome references, because what this software does is analyze all of the available reference genomes, and it selects marker genes from those that are informative for taxonomic purposes. So if you use this software, you don't actually end up using, let's say, 20,000 genomes, which is what you would be having to use otherwise. You just use very small informative selections from those, because you have large proportions of the genomes that are just not informative because they're shared across too many different species. So they have used high performance computers to do phylogenetic analysis of tens of thousands of genomes and select the parts of those genomes that are useful for taxonomic classification. And the software provides its own reference database. And if you use Metaflan to analyze your profiles, and this is very, very widely used software, the first thing that will happen is it will automatically download the most recent reference database, which is about 15 gigabytes, I believe, and that's just for those marker genes, and then it will use those for taxonomic profiling. It would be an enormous challenge to actually go back to all of those genome references and analyze them manually, and there's no point in doing it. Metaflan also analyzes one profile in a matter of like minutes, even on a regular laptop. So because of the reduced size of the reference genome sequences, and because of their specificity to species and difference between one another, this mapping occurs very quickly. And it's general. You can use that for all kinds of human microbiome studies. I should say, you know, since it depends on reference databases, it's going to be most accurate for human microbiome studies, especially for stool. There are not very many unknown species in human stool anymore, but if you're analyzing environmental samples from the forest or something, it's going to be less effective because there's less coverage of those microbial genomes in the known reference databases. And then if you're analyzing more rare microbiomes like that, might have to do some metagenomic assembly to identify what is there, which is a whole different kind of analysis. Does that answer your question?
Okay. There is one question more.
Yes, analysis of disease in animals. I have not, but I have seen plenty of papers like this. The analysis is the same. The species and the definitely the genera that they're mostly the same. You can use the same software and you perform differential abundance analysis and these different things the same way as you would in humans. So it's not like genome analysis where you need a different reference genome for studying an animal other than human. We can use the same reference genomes. It might, if depending on the animal, like certainly if it were sea cucumbers, it's probably going to have a very different microbiome than humans, and we might have poor coverage of their reference of the microbial genomes. But if it's bovine or mouse, there's going to be pretty good coverage of those microbial genomes.
Okay, I got a message from my student. I think he is afraid to ask you because he said that I am from statistics and most of the material in here is not about statistical method. Would you like to, to suggest to the participant, which is undergraduate student from statistics, what should they do if they would like to do like microbiome analysis? Thank you very much.
Like, I guess the question is about background reading for learning about statistical analysis of microbiome data.
Yes, they would like to study or do the research as like you. What should they prepare? Something like that.
Yeah, there's not like I guess a textbook that I could recommend that is specific to microbiome studies. There are a number of review papers that I might recommend, and also benchmarking papers that would be good places to start that would review some of these methods. I'd be happy to provide those, to just provide a list of some references for someone from statistics wanting to analyze microbiome data that introduce the statistical challenges specific to microbiome data. This paper in genome biology would be not a bad, it would definitely be part of my reading list.
Mm-hmm.
But there are some other ones that I would give. This one is more of a benchmarking paper that does introduce some of the statistical issues, but it's more of it's an analysis. It's not focused on giving an introduction to the field. There are some other review papers that are more of an introduction.
Okay. Now my question: do they need a course like molecular biology or something like that before doing the analysis?
I don't think so.
No?
I think many biostatisticians who have never taken a course in molecular biology analyze microbiome data just using the tools that are available and rely on their collaborators to interpret the data. Well, not to go back to promoting my own work so much too much, but just reading some microbiome papers and trying to distill the essential information from them as captured by BugSigDB is also a good way to learn about the field and the methods that are being used for analysis. These pieces of information that it collects are based on my discussion with a lot of people about what are the most essential components of a microbiome study that we need to understand to assess a study. And we've created a controlled vocabulary for describing these things. But yeah, again, there are different approaches to introducing yourself to a field. And I have a lot of master's students on this project who don't know anything about biology or about the human microbiome. I have a set of papers that talks a bit about the microbiome at the level of like my first few slides, like what is the microbiome, what is the taxonomy, what is alpha diversity, questions like this, and then some that are what are the methods that people usually use for analysis. But these are all kind of picked out by me because it's a relatively new field, and I think, and it also changes quickly, so it's difficult to write a textbook that's going to remain relevant for very long. But I'm a big fan of learning by doing, of by reading the literature and just trying to do your project.
Okay. Thank you very much. Any other question? Bu Dina, Pak Hasan, or Pak Muhajir? No? Okay. For me, the talk is really interesting, even I'm from statistics in background. Some of the participants in here is coming from India, and also they are working on bioinformatics, but my student is not working in bioinformatics. I would like to highlight from statistics view. The first one is doing research about microbiome or bioinformatics. It means you are dealing with data which is about high dimensional data. Okay. And then the second one, you don't have to master doing experiment to produce the data, but usually the data already been provided by people from wet lab, something like that. You need to understand about the question or the problem related to that data. And then you have to choose the right analysis, maybe including something like, for example, visualization, because it is involved more visualization in data from bioinformatics. And then after that, you can do the conclusion, answering the question of the problem. That's from statistics point of view, because I'm worried maybe my student a little bit confused why this is, for example, talking about microbiome, but actually would like to have concentration on bioinformatics in the future. So I think this is a highlight for a new student, which is year 2020, academic year 2020 and 2021. I think this is a very good introduction for you. And Professor Warden also told you that you don't have to be mastering about biological thing. I think you can read some paper. It is more than enough, which is research from other experts. But if you want to be an expert, I think you have to go more deeper. Okay, thank you very much, Professor, for your talk and also for the participants. And then I would like to give the time to the Master of Ceremony, Mbak Zahra. The time is yours.
Okay. Thank you to Dr. Rahmatul Fajriah, MSc, for moderating our webinar today. And of course, thank you to Dr. Levi Waldron for sharing with us. It is such a great opportunity and our pleasure to have you with us. But before, probably, we will give you symbolically the certificate of appreciation. So probably, yes.
he would like to show you the certificate of your talk, but okay.
Yeah.
I think it is not yet.
Okay.
Okay. That's your certificate, Professor.
Thank you.
Okay, here is it, the certificate of appreciation for Dr. Levi Waldron as an associate professor in Department of Epidemiology and Biostatistics, the City University of New York. It is the form of our appreciation because we thank you very much for sharing the great knowledge with us. I'm sure that it is will be beneficial personally for me and of course for all the participants. Once again, thank you so much.
Thank you very much. Thank you for all of the great questions, as to my hosts and to the participants for their attention and questions.
Okay, you are welcome.
Okay, so ladies and gentlemen, before we end this event, I'm gonna also show you first, give the announcement about our upcoming webinar. So to the committee, can you show the information about our upcoming webinar, please? Okay, here is it, our upcoming webinar, which is SBRC Webinar Series 6 with the speaker, Mr. Ricardas Ditikis, the Associate Professor of University of Western Ontario. And it will be held on 28th August 2021. So for information, the topic is about
He will talk about statistics and about actuarial science. Yeah.
Thank you. So for those who are interested, we can't wait for your coming in our upcoming webinar. And also don't forget to the participants to fill the attendance form because it will be used for the certificate. So, well, ladies and gentlemen, now we come to the end of this SBRC Webinar Series 5 2021. Hopefully, this webinar will be beneficial for everybody. On behalf of the host and committee, we would like to extend our deepest appreciation to the speaker and to all participants for your support and participation. So please give applause to our speaker and also to all of you. Once again, I'm, as the Master of Ceremony, say thank you for everyone for your participation. Good evening, stay healthy, and see you on the next webinar. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you,
Probably
Wadron. We wish you maybe we can join also our